recording. Okay, it's good. Oh, that's about right. Oh yeah, it's only good. So everybody can hear me.
Yeah, we can hear you.
Good. I got to turn my sound down. It's too loud. Okay, so ready to do the enzyme lab virtually. All right. So I'm gonna do let's talk about enzymes, how they work. This is what I did in lab yesterday. We talked about how they work. And then we went ahead and did an experiment on the effect of temperature on enzyme activity. And fortunately, Ms. Boward, who is one of the other instructors in for Bio 106, she joined my lab and she took photographs and put together a video of doing the experiment. And she also did a PowerPoint, which I'm going to go over at the end. And then once we get the data for the whole experiment, so we're going to use the data that we got yesterday. It was pretty good data. Oh, somebody in the waiting room. We'll go over the questions that you need to answer on the lab document. Actually, let me show you the lab document. Let's see. Here we go. So here's the lab document, and this gave a whole. So I'm going to go over this stuff first, and then at the end, it gives you all the instructions on how we did the experiment. And then at the end, you write in the data. I'm going to give you data to put on this analysis of results, the data that we got yesterday. And we got to answer this question up here. And then we answer these questions here. And then you turn this into me and I grade it and you get points. Oh, somebody's in the waiting room. So that's what we're going to do. And if there's any question along the way, like I said, interrupt me. All right. If you got any question about anything. Okay. A lot of lab documents have this getting ready thing. It's like a pre-lab thing. It's good to go over it, but I don't expect you to do that part, fill in that part. I usually go over it myself. You fill in the back part. So like getting ready number one, diagram an amino acid. We don't do that. We already went over amino acids. Condensation reaction is the same as the dehydration reaction. We put them together. List the 20 amino acids. No, you don't have to list 20 amino acids. How do they differ? Remember, by the R group. They all have different R groups. Okay. Draw a picture of a primary, secondary, tertiary, and quaternary protein. No. Primary sequence, remember, is the order of amino acids in the protein. Secondary and tertiary are how these amino acids that attach to each other form a three-dimensional shape, all right, molecular shape. And I can't emphasize how important molecular shape is. And they're usually held together by hydrogen bonds within the molecule itself. The amino acids that make up the protein are bonding to each other with hydrogen bonds, and that gives it the three-dimensional shape. Right? All proteins have primary, secondary, and tertiary uh, structure. Some have what's called quaternary, where you have more than one polypeptide chain that are interacting together, like hemoglobin, the protein that carries oxygen in your red blood cells. It has quaternary structure. It's 4 polypeptide chains interacting. It's a huge protein, 453 amino acids long. So they don't all have quaternary. Okay. Now define the following terms, number 4 here. Okay, enzyme. Um, First we talk about catalyst. You gotta know what a catalyst is. A catalyst is a molecule or something that speeds up chemical reactions without being used up in the process. Right? Um, and uh, and I give the example, a real life example of what it's of a inorganic catalyst in your car has a catalytic converter. Back in the old days, cars didn't have catalytic converters, and they put out CO2 and water, but they also put out these burnt hydrocarbons that was really polluting. Now we have a catalytic converter. It converts those unburned hydrocarbons into CO2 and water, and that's the only thing that comes out of your tailpipe now. CO2 is a greenhouse gas, okay, but it's a lot better than those unburned hydrocarbons. And the thing is, you never have to replace your catalytic converter, do you? It lasts forever because these can be reused over and over and over again. That's an inorganic catalyst. Enzymes are organic catalysts, right? And they're absolutely essential for life. Why? Well, a lot of the reactions that are going on inside your cells, are what's known as spontaneous. They go all by themselves. Even the reaction in lab that we were studying, the breakdown of hydrogen peroxide into water and oxygen, happens all by itself, but very slowly. So the thing is, remember, life is a really fast-paced enterprise. Things have to be fast. Like we saw in the water lab, diffusion. That's why cells are small. Diffusion has to be fast. And if you had really big cells, it wouldn't be. So we need to speed up chemical reactions, and we do that with enzymes. And so they're absolutely, so every living thing on Earth has got enzymes, and they're absolutely critical for life. Okay, there used to be RNA molecules back in like a couple billion years ago, or several billion years ago. They were actually RNAs, but they were taken over by proteins, except for one. Later on, when we talk about protein synthesis, we'll talk about an RNA enzyme. that's still around. It's a really ancient enzyme that's still here, but all the rest have been taken over by proteins. Okay, so we know what a protein is: amino acid, amino group, carboxyl. We went over this in the molecular models lab: dehydration, synthesis, peptide bond, primary, secondary, tertiary structure. Just mention it. Quaternary structure, denature. That's something you have to define. We're going to see that temperature affects enzyme activity, and that's what we're going to test today. How does temperature affect enzyme activity? Um, and so we're going to do this experiment. We had 3 tables, and one table did it at 2 to 5 degrees in ice water. One table did it at room temperature, which was about 22 to 24 Celsius. And one table was a hot table, 60 to 70 degrees. So it heated up water on a hot plate. And enzymes will work at all those temperatures. But if you bring an enzyme, or actually you bring any protein, to 100 degrees, well, regular protein, you bring a protein to 100 degrees C or 212 degrees Fahrenheit, boiling. You boil a protein, that's called denaturation. You will denature the protein. That is, you will irreversibly destroy its three-dimensional structure. You cool it down, it'll never work again. It's gone. So what you've done when you boil it is you destroy the quaternary. If you had quaternary, the tertiary and secondary structure, you've destroyed it. You didn't destroy the primary structure. You still got the amino acids in this, but the three-dimensional shape is gone and won't come back. That's why boiling anything, any living thing, is going to kill it. Except, there's always an exception to stuff. Remember the archaebacteria. You can go to Yellowstone National Park, look in these boiling hot springs, and it's like green mats in the boiling water. These are archaebacteria. They can survive boiling water. They're like the only things, because their proteins are really structured differently. So they're a bizarre exception. Okay. A chemical reaction, a substrate, active site, product, activation energy. So let me define those by talking about how an enzyme works. Oh, somebody's in the waiting room. Some people getting bumped and put back in the waiting room for some reason. All right. Let me stop sharing this and share. Excuse me? Okay. Okay, Adrian's back in. Let me share something else with this drawing I put up on the board. This thing. Okay, I drew this crude drawing up on the board. All right. This is a chemical reaction. A is converted to B and C, right? And the thing to the left of the arrow, if this was a non-enzymatically catalyzed reaction, that would be called a reactant on the left side of the arrow. Right? But an enzyme catalyzed reaction, that's called a substrate, right? And then the enzyme is right here, and we'll be testing an enzyme called catalase. And here are the products. So this is a degradation reaction. A is broken up into B and C. Sometimes it's a build-up reaction. You know, B and C could be made into A. So it could be multiple substrates or multiple products or single substrates, single products, whatever. So there's the products, B and C. Okay, so there's two ways you can look at see how enzymes work. One is this way right here. This is, think of this in 3D. You can't draw in 3D, but think of it in 3D. It's a three-dimensional thing. And the shape is really important, particularly the shape of what's called the active site right here. Because this is where the substrate is going to fit into the active site. So you have complementary shapes where they fit together. Sometimes called a induced fit or lock and key mechanism. Like how do keys work in locks? Well, they have a particular shape, right? Well, you put it in the lock, and the lock has a complementary shape. They fit together, and you can turn and unlock the lock. Right? Wrong shape, key with different shape, not going to turn the lock. Same thing with enzymes. So you have to form what's called an enzyme substrate complex. And then B and C are split. A is split into B and C. B and C take off, diffuse away, and then the enzyme comes right back, has an empty active site, finds another molecule A, breaks it to B and C, goes back and back, around and around and around, and like I said, doesn't get used up, doesn't get changed in any way in a reaction. Can be reused over and over and over again. And the speed at which it does this is called a turnover rate. And the catalase, the enzyme, were we tested in lab, it's got a very high turnover rate. Now, eventually, proteins break down. Okay, so eventually it's going to break down. And then you have to remake it. Then you have to make the enzyme by going into a gene and doing protein synthesis and making the protein. So anyway, so that's one way you can look at see how enzymes work. The other way is down here, talking about what's called activation energy. Wait a minute. Somebody trying to get in. Hang on a second. No. Okay. So you make up this graph. Energy on the y-axis, time on the x-axis. Every molecule has got a certain amount of energy in it. It doesn't matter what it is, because every molecule is held together with bonds that are electrons, and electrons are pure energy. Okay, so you can have here you can say that A has got a fair amount of energy in it. B and C have less energy in it, and that would be a spontaneous reaction. If we had a build-up reaction, B and C made into A, you may have to use cellular energy to make that go. But this is a spontaneous reaction going from a high energy to a lower energy. Okay. So how does it get from A to B? Does it go straight like this? No. And here's the energy released by the reaction. Difference between A here and B here. Okay, so even really spontaneous reactions and even actually explosive reactions, they just don't go all by themselves, usually. For example, I always give the example of in the room we have these gas jets on the tables, right? Let's close the doors, turn on all the gas jets. Let's fill up the room with natural gas in an oxygen atmosphere. Good idea? No. But just doing that, will the room blow up? I think it would. Just by doing that. You want to answer it? No? Well, answer is no, it won't blow up. But all we needed was a little tiny spark, even a static electricity spark, and you blow the whole room up. That's the activation energy. That would have a very, very small activation. So every reaction, you have to add a little energy to it to get it started. That's the activation energy right here. And it's a very, very good thing that the world works this way. Because everything in the room, well, everything staying around you is all combustible. Everything's combustible. But why does this paper over here burst into flame? Because we're in an oxygen atmosphere, and of course you need oxygen to burn stuff, right? You can't burn stuff in an oxygen-free atmosphere. So it's in an oxygen atmosphere, and it's combustible. It doesn't burst into flame because the activation energy is too high. Right? But I set a match to it, I can burn everything down. But some things have a very low activation energy. Like you bring in an open, container of gasoline into the room. Not a good idea either. But will it burst into flame? No. But it has a much less activation energy. So occasionally, if you have something that really has a low activation energy, just spontaneously these molecules could find a way over that hump, and that's spontaneous combustion. And it's a low possibility, but it still is. So that's why they tell you don't leave open containers of gasoline or gas-soaked rags in your garage or something, because there's a slight chance that they can they can reach activation energy all on their own. Okay, so without the enzyme, this is what you do. You got to add something to get this thing going. With the enzyme, though, it's this curve. It lowers the activation energy greatly. So it goes really fast and spontaneous. All right? So everybody understand that?
Yeah.
Okay, good. Okay, let me stop sharing this and go back to the paper. Okay, so here's this enzyme plus substrate, enzyme substrate complex, enzyme product. Here's the activation energy, the thing that I crudely drew, right? Okay. Like I said, could biochemical reactions occur without enzymes? Yes, but too slowly. In this lab, we're working with an enzyme catalase. All cells contain this important enzyme that transforms hydrogen peroxide, H2O2, into water and O2. Here's the reaction down here. This is a really toxic molecule, hydrogen peroxide. Well, as I mentioned in class, you probably have some, you might have some at home. What do you use it for? Give you a hint, it's probably in your medicine cabinet. Anybody?
Cuts.
Cuts, right. So what do? You pour it into a cut, right? Why?
Doesn't it like clean it out?
Yeah, but how does it clean it out? What does it do? Well, when you cut yourself, what gets into your cut? Think living things.
Bacteria.
Bacteria. You're covered with bacteria. You cut yourself, bacteria gets into the. So you want to clean that out. So what does the catalase do? I mean, what does the hydrogen peroxide do? Is it kills bacteria. So bacteria are cells, right? So the take-home message is it kills cells, all cells. You're gonna kill some of your own cells when you pour it into your wound, but we make that cells when your wound heals. You replenish those cells. So it's a really dangerous molecule. Now here's a thing. When you pour it into the cut, what do you see?
Doesn't it like fizz?
It fizzes. Yeah, it fizzes. And that's because you have catalase in all of your cells. You cut yourself open, you break open cells. The catalase falls out of the cells into the open wound. And when you pour the hydrogen peroxide in, your own catalase breaks it into water and O2, which is where the bubbles come from. So at the same time, it's the hydrogen peroxide is killing the bacteria, your own catalase is converting it to water and O2. All right. And that's why it fizzes. So, and here's the thing. Cells cannot help but make this molecule. They don't want to make it, but they do. It has to do with the metabolism of all cells on Earth. They can't help but make this molecule. And it's a really dangerous molecule. So they need protection against it. So they have catalase and they have it on hand. So the instant they make the hydrogen peroxide in their metabolic processes, they destroy it. So to protect themselves. All right. So all cells on Earth pretty much have catalase, right? And we're gonna and it's same too with potato cells. That's where we're gonna get our catalase, from potatoes. So you would have been peeling potatoes in lab. That's the first part. Okay. Okay, so what we want to do is measure the rate of this reaction. You know, and how do we do that? Well, we measure the amount of the products that are made. It would be pretty tough to measure how much water is made by this reaction, but we can measure the amount of oxygen made in this reaction. So we're going to look at the enzyme activity of catalase as a function of temperature. So temperature on the x-axis, enzyme activity on the y-axis, and what's the measure of enzyme activity is how much oxygen is produced. The more oxygen produced, the greater the enzyme activity. The less oxygen produced, the less activity. So that's what we're going to directly measure. How much oxygen, milliliters of oxygen produced per minute. All right, so we start off by preparing the enzyme extract, obtain, peel, and chop a medium-sized potato. They weighed it out, 50 grams. This is a group activity. So we had three tables, as I mentioned. A low temperature table, a room temperature table, a high temperature table. And let me, rather than talk through these procedures, I'm going to show you the video that, let me share that. I'm going to look at a video made by Miss Boward. At least I hope so. Let me see if I can share that screen and play that video. Okay, can I share this screen? Yeah, here we go. Okay, this is going to work, I think. All right, so we'll look at this. So potatoes peeled and chopped, put in the weighing boat, 50 grams. I don't know why she put the music in the background though. This was yesterday in section 01. Then they add the potato and 100 mils of water to a blender to make a potato slurpee or smoothie, frappe. Yeah, I'm gonna turn the music down. 2 filtering steps, one through cheesecloth, and the next one is called the Buchner funnel. I can full screen this. Yeah. And this is called a Buchner funnel, second filtering step. Well, the eudiometer. Let me pause this thing. This thing right here, it's called the eudiometer. And it's graded, and you fill it with water, and you put the open end of it into a tray, trough of water, right? And you up here you note where you start, and you get a number of milliliters where you start, and that's crucial. Okay, let's continue. So you measure 15 milliliters of potato extract and put it into a reaction tube, which is a large test tube. They got to keep it on ice because it's a protein and bacteria are getting into it and they're gonna chew it up. So you got to keep it. If you're not the extract you're not using, you got to keep it on ice. Or you should keep it on ice. So you put the test tube with the enzyme extract in a beaker of the particular temperature you're using to test the enzyme activity. This happened to be the hot temperature group. They were putting theirs at heating up water to 60 to 70 degrees. And you got to put the enzyme extract in there for about 5 minutes and use the same temperature throughout the length of the experiment. Okay, here, if you see this thing right here, this is a plug here that's going to be put in the top of the reaction vessel, and in that cork is a glass tube connected to a rubber tube, and it has a top of a syringe. Now they twisted off the syringe and are filling it with 4 milliliters of hydrogen peroxide, the substrate. Okay, here's the setup. So here's the enzyme extract down here. Here's the substrate, the hydrogen peroxide. She's about to inject it into the enzyme extract. And what's going to happen? You're going to make water and oxygen. The oxygen will travel up the tube, up the reaction vessel, into this tube, down through here, and up into the udiometer. And so the water level in the udiometer will drop depending upon how much oxygen is going into it. All right. So you're collecting the oxygen in the udiometer. Oh, so once the hydrogen peroxide is injected, they twirl it once, they leave it in the same temperature, and it's only a 2-minute experiment. Do all this for 2 minutes. And after 2 minutes, you go over the udiometer and you measure how far the water is dropped, and you get a new reading below. And you subtract, so you write that down, and you subtract where you started from where you ended. And that difference is how much oxygen was produced by this reaction in 2 minutes. And you divide that by 2 to get oxygen produced, milliliters of oxygen produced per minute, which you're going to do here. Okay. Wait a minute, let's stop this now. How do we get out of this? Exit full screen. Okay. And they did that 3 times. 3 times at their particular temperature, right? And then and they get a three totals for how many milliliters are produced per minute. They added them up, divided by 3 to get an average, and that average we put up on the board. No, wait a minute. Where's go back to the. So these are all the instructions here, and here where they wrote down the data. Starting volume was the millimeters of water in the udiometer when you started. Ending volume was after the 2 minutes. What did it drop down to? Net volume, subtract the starting volume from the ending volume. Temperature was either 2 to 5, 22 to 24, or 60 to 70. Time elapsed was all the same, 2 minutes. And this was milliliters of oxygen produced per minute, is this number divided by 2. And then you add them all up, divide by 3 to get this number. And then we put up the data up on a board, the 3 averages up on a board, and down here you would write the average for the 2 temperatures your group didn't do. Right? Now, and I'm going to show you a short PowerPoint also that to emphasize the same thing that Ms. Bauer put together. It's going to be a lot of saying the same thing, but when I give you the data at the end here that was actually collected yesterday. So we're going to look at the what we found out. What is the effect of temperature on enzyme activity, and why does that happen? So let me share that screen. Okay. Okay, so we can go over this quickly. We've already gone over this catalytic converter. It's a good thing you don't have to replace your catalytic converter because it's really expensive. I had to replace mine once because I lost it, ended up on the road someplace, and it's really expensive because it's some kind of platinum or palladium catalyst inside, so it's expensive. Enzyme. Okay, so it's enzyme structure: primary structure, secondary structure, tertiary, held together by hydrogen bonds, and some of quaternary. Okay. Same thing here. Mechanism of enzyme activity. Enzyme substrate complex. Products come off. Enzyme goes back and does it. Everything. There's the activation energy, and this is available to you in on your D2L lab site. It's made available. Oh, so factors affecting enzyme activity: temperature, pH, enzyme concentration, and substrate concentration. We don't have to. So pH works in the same way that pH also affects enzyme activity, and we're going to see why that is in a little bit when we talk about the results. Denaturing. Remember, you boil it. It's gone forever. If you just heat it up a little bit and you bring it back down again, it's fine. It'll work, but not if you boil it. That's true for real high temperatures and really extremes of pH also. Really acid, really base. So the proteins that you eat, for example, are denatured when they get to your stomach because your stomach pH is around 1.5 or something, and it just denatures any proteins that go into it. Okay, here's catalase, and this is the reaction. Now, this is what's called a balanced equation. You're never going to have to balance equations in this class. But what's important if I put up reactions is what's over here to the left of the arrow and what's over here to the right of the arrow. And the important thing to remember is you're transforming energy and matter. You're not making energy or losing energy or making matter or losing matter. So if you got X number of atoms on this side, you're going to have X number of atoms on this side. And that's called balancing the equation. But we don't have to worry about that. You can, if you want to learn how to balance the equation, you can take a chem class. Okay. Okay. So what temperature is optimal for catalytic activity? All right. And we measured enzyme activity as a production of O2. So about 4 degrees room temperature and 60 to 65. Okay. There's the setup that we used to collect the gas. Okay, here's the data that we got. Cold, 9.7 milliliters per minute. Room temperature, 13.52 milliliters per minute. The hot, 2.65 millimeters per minute. So you're supposed to graph it. It says in the lab, oh, use a computer to draw a graph. You only got 3 data points. You don't have to do that. Make a thumbnail. Just draw it freehand, make a thumbnail. Whoops, somebody's in the way of me. A thumbnail, and what does a graph look like? It's a bell-shaped curve. Does it have, okay, let me go back to, hang on a second, let me go back to here. And it shows a bell-shaped curve, actually. Right here, factors that affect enzyme activity. Here, bell-shaped curve. There's an optimal temperature. Like all enzymes have an optimal temperature where they work best. Right? And it falls off on the low side and falls off on the high side, but for different reasons. Now you might have we used to in the lab document we used to have students make a hypothesis as to how do you think temperature is going to affect enzymes? And most people would say, well, the enzyme activity is going to be greater at a higher temperature than a real low temperature, like between ice water and room temperature. Enzyme activity is going to go up from ice water to room temperature. Why? Well, remember what temperature measures: molecular movement. That's what temperature measures. And enzyme activity is dependent upon two molecules hitting each other in the right way, right? The substrate hitting the enzyme in the active site and forming the enzyme substrate complex. So at a cold temperature, molecules are moving sluggishly, slowly, and they hit together less often, more slowly. You raise the temperature, molecules move faster and they hit faster, and the enzyme activity goes up. So that's why it was higher at room temperature than ice water. Okay, but why did it go down at 60 or 70? Because if you graft if you graft products produced in a regular chemical reaction, not enzymatic reaction, but a regular chemical reaction, um, you'll see that the curve of products produced as a function of temperature is a straight line with a positive slope up. The hotter you make it, the faster you make products for regular chemical reactions, but not for enzymatically catalyzed reactions. It goes down after you go past the optimal temperature. But why? It's for a different reason. Remember back in the water lab, we talked about how many hydrogen bonds are produced as a function of temperature. Remember, that was a negative slope. The higher the temperature, the fewer hydrogen bonds. So what holds together a protein in a three-dimensional shape? Hydrogen bonds. So you heat up to 60 or 70, you are destroying hydrogen bonds, and therefore you're altering the secondary and tertiary structure. So you're changing the shape of the active site because it's held together by hydrogen bonds. And you change the shape of the active site, the substrate's not going to be able to fit as well, and that's going to lower enzyme activity, how much products are produced. So it falls off on the high end for a different reason. You're changing the shape of the active site. Everybody see that? Okay, remember pH, bell-shaped curve also. Enzymes have an optimal pH, they have an optimal temperature. You don't have to worry about these guys on the enzyme concentration and substrate concentration. Those are bell-shaped curves. And so then I would say, well, what do you think your optimal temperature for your personal catalase is that's in your cells? What do you think its optimal temperature is? Anybody? What temperature is it at?
Would it be like the temperature of our body?
Which is what?
96 or 97.
Well,
Right.
that's Fahrenheit, but in Celsius, it's actually 37. Your body temperature is 37 degrees Celsius. And so that's your optimal temperature for your catalase. But potato, what's a potato's body temperature? Well, whatever, it's room temperature. So that's why we express. But If we want to look at get an optimal temperature for a human catalyst, we'd have to heat up a little bit to 37 from 22, 24. And same thing with pH. Our enzymes work best at 7.4. And like that's why body temperature and pH pH of your body and temperature of your body is regulated to maintain molecular shape, to make sure your enzymes don't change shape so that they work. Because if they don't work, you don't work, right? So that's why those things are regulated. Okay. Now, need to write down this data here. We haven't written it down yet, right down, because it has to go on to your Rob document that you turned to me. All right, we need to use that data. Right. Everybody got that? Right. Okay, so now we're just going to answer the questions, and then we'll be all done. Okay. So the first question here, determining a temperature optimum for catalase enzyme from potato. Why was it necessary to use a syringe to add the hydrogen peroxide rather than to remove the stopper and pipette the hydrogen peroxide directly into the generator tube? In other words, you got this sealed system. Why not just have the enzyme activity and just pipette in the hydrogen peroxide and then close it and then capture the oxygen? Well, you would lose a lot of the oxygen that was produced by the reaction if you did that, because it's a really quick reaction. As I said, catalase has a high turnover rate, and no matter how fast you try to put it in and close it up, you'd lose some of the oxygen. So you have to have a closed system to make sure that all the oxygen goes into that tube, up the UV and drops the level to collect all the oxygen. That's why. All right. So you have to put in here, because we didn't actually do the experiment. But make sure you write down the three temperatures here and the milliliters of oxygen produced per minute for those three temperatures with the data that we collected yesterday. Write that down here, right? Okay. Then, number 3 says, using a computer plotted column graph using the data above. Choose an appropriate title for your graph. No, you don't have to use a computer. We only have 3 data points. So, draw a thumbnail sketch like you did in the water lab. Enzyme activity measured by milliliters of oxygen produced per minute as a function of temperature. And put the 3 dots on, draw the bell-shaped curve, and that's fine. Okay. And number 4, observe your graph. Determine the temperature optimum for a potato catalase. And of course, it's room temperature, 22 to 24 degrees C. Explain how and why the enzyme works at low room and high temperatures in the figure legend. We really have to, what you should put in there is, why is it lower at low temperature? Why is it lower at high temperature? Okay. Remember, it's lower at low temperature because molecular collisions are fewer and farther between. The molecules are moving much more slowly. They collide more slowly, and so they make less oxygen. They make less product per minute. Okay. Than they do at room temperature. Then remember, on the high side, the higher temperature is breaking hydrogen bonds and breaking down the secondary and tertiary structure. In other words, changing the shape of the active site. And therefore, substrate doesn't fit as well. You make less product, less oxygen. All right, 5, right? write out the equation. The equation is right there. 2H2O2 arrow 2H2O plus O2. And then name the enzyme, substrate, and products in this reaction. So why don't we do that right now? What's the name of the enzyme? Somebody come up with it. Anybody? What's the enzyme name? Let's go back. Here's what you got to do right here on this page here. So what's the enzyme name? Tells you right there.
Catalase.
Catalase is the. Okay. Name the substrate. Remember what the substrate is, the thing the enzyme acts upon. What's that in this reaction?
Is it hydrogen peroxide?
Hydrogen peroxide, that's the substrate. So the products are what? What did you measure? To the right of the arrow.
So would its products be water and oxygen?
Those are the products. That's it. So that's straightforward. Now see, you're gonna need to know these enzyme terminology. It's gonna be on the next exam. So you gotta know this. You gotta know how enzymes work in the enzyme terminology. Right? I think this is in chapter 5, the working cell. Finally, question 6. Why do all tissues make this enzyme? Well, it's a protection. Because remember, all cells make hydrogen peroxide and they can't help themselves. It's a natural byproduct of metabolism. And it's a very dangerous molecule that actually kills cells. So they have to have catalase on hand to destroy it as soon as it's made to protect themselves. Let me end this show. So stop sharing. Oh, there's somebody in the waiting room. Oh no. Adrian, kicked out again. I know why you're always being kicked out there, Adrian. Okay, so I'm recording this. So if you want to see it again, I'll put it up on YouTube site if you ever want to get back and you need to see it again, right? And I had it set to automatically record to make sure I didn't forget. So any questions, thoughts, problems? If not, I'm gonna end the meeting in a minute or so if there's no questions.
Okay, back at question 2, where it's trial 1, trial 1, trial 3. All the the
Yeah.
starting volume, the ending volume, was all that in the video?
Well, they showed them. Remember, there was a student peering at the UDiometer before the experiment started, getting the starting volume in milliliters. The UDiometer is graded in milliliters, you know, and it was each milliliter was certain, and it was 5 lines between each millimeter, so each line was 2/10 of a milliliter. So you'd be like 7.8. You start at 7.8 milliliters. You write that down, starting point, and then you do the experiment. The water level drops because the oxygen's coming in and replacing it. And let's say it goes down to, it started at 7.8, it goes down to 27.8. It goes higher numbers as you go down these numbers. So then you subtract, to make it easy, I mean 27.8. So you subtract starting point 7.8 from 27.8, where it ended up after the 2 minutes, and you get 20 milliliters. So over the 2 minutes, the reaction produced 20 milliliters of oxygen. You write that down, you divide it by 2, because that's in 2 minutes, and you want milliliters per minute. Divide it in 2, and that will be 10 milliliters per minute produced by the reaction for that trial. Then you do it again, and say maybe you get 12 milliliters produced in the second trial. Maybe you get 8 produced in the third trial, and you add up all those, take a mean, and that would be the number that went up on the board. See?
Okay, I see what you mean.
Yeah. Yeah, this is a good experiment. It almost always works out to be that nice bell-shaped curve. Because well, we used to have problems with that because I forgot that the room temperature group has to keep their I usually had to tell the room temperature group, you guys don't have to do anything, just put it in and run it. But then I didn't forgot that they were keeping it on ice prior to doing the experiment, and so if they would have to warm it up to room temperature, so put their hands around it, warmed it up to room temperature, and then did the experiment. Otherwise, they get the same numbers as the ice water group. Anything else? So you can send it to me an electronic copy if you want filled out, or you just hang on to it when you come back to the campus. You guys will be coming back to campus the week after next to do the next on-campus lab, unless it's snowing again. Remember, and then next week you do the at-home, the cells lab at home, and that's just a series of reading about cells and filling in blanks. And somebody's say, can we do a lab on that? Remember, we have Zoom meetings, regularly scheduled Zoom meetings every Monday morning at 10 and every Tuesday morning at 10. So if you need help with that lab, just come into one of those Zoom meetings, and you can ask me questions about that if you're having trouble filling that out. And I think next week is time for exam 2, I think. It's going to be on chapters 4 and 5, a tour to cell, and a working cell. And I'll be putting that together soon. And I opened up that chapters 4 and 5 in Mastering Biology at the Pearson website too. So I think that's all my announcements. Okay, I'm gonna end the meeting in a minute or so, unless there's other questions or comments or problems. Okay, that's it. See you everybody. Stay safe, stay warm, and see you again soon. Hope. Okay. Bye bye.